frequent bacteria found in Indonesia, resistant bacteria, and most frequently found in Indonesia, from a very expert professor from Australia, namely Professor Alison Kesson. First, I would like to greet Professor Alison. Hello, Professor Alison. Good morning. How are you?
Good morning. How are you? Well, it's good afternoon here, but good afternoon.
Yes, it's very good for me also to see you again, Professor, and also a great happiness for us to listen from you about the topic which is rarely discussed in Indonesia, because maybe only few doctors understand about this. But actually, the problem is already very big. Big in very frequently found in our clinical practice. So, but before you start the presentation, please allow me to introduce you to the audience. So I would like to share. Can I share the screen to the sharing set? Okay, so Professor Alison Margaret Kesson is from Australia. He works in the Department of Infectious Diseases and Microbiology at the Children's Hospital at Westmead, Sydney, Australia. She has her educational background is that she already graduated from Bachelor of Medicine and Bachelor of Surgery in 1978, and then postgraduate at Royal Australia College of Physicians, Australian National University, and also College of the Pathologists Australia. She also a very active scientist, with joining Australia Society for HIV Medicine, Australia Society for Infectious Diseases, and also Australian American Society for Microbiology. She has a long extraordinary appointment from 1978 until 2023, and also a lot of publication, scientific work in publication, editorship, presentation, research administration, journal reviewer, and research grants, and also professional development. And she also has a long list of administrative position, like chair of division of diagnostic services in Children's Hospital at Westmead, Sydney, Australia, head of antimicrobial stewardship program, and also in the Sydney Hospital, Children's Hospital, also the chair of the Infection Control Committee of the Children's Hospital at Westmead, and also head of infectious diseases and microbiology at the Children's Hospital at Westmead, and also the head of pathology, the Children's Hospital at Westmead, also the head of virology and microbiology, the Children's Hospital at Westmead, head of virology laboratory, Hunter Area Pathology Service, and other position, which is very completely give opportunity for him to understand about the topic that we are going to discuss today. So now I would like to invite Professor Alison Kesson to start the presentation. Your time will be 45 minutes, and then we will discuss for another 30 minutes. Thank you. Professor Alison, the time is yours. The screen is yours.
Okay. Okay, so it's there. Okay, so you've asked me to talk about CPE infections, carbapenemase producing infections, so diagnosis, treatment and prevention. But I'm going to start with a little bit talking about other forms of resistance and you can see how this has evolved over time. And so you will see it in the sort of big picture, and then we'll talk about the carbapenemase. So beta-lactams. Beta-lactams are antibiotics that have a beta-lactam ring nucleus, and this is a picture of it over here. This is what it looks like. And beta-lactams include penicillins, cephalosporins, kefamycins, such as cefoxidan and cefotetan, Carbapenems, which is imipenem, meropenem, ertapenem, and Amonobactam aztreonum. So they're the beta-lactam group. So beta-lactams exhibit antibiotic properties by inhibiting bacterial cell wall biosynthesis and target many gram-positive and gram-negative bacteria. Bacterial resistance occurs as a resulting from production of many beta-lactamases, which are enzymes that break open the beta-lactam ring and stop it from being the drug from being effective. Now, there are more than 1,800 different beta-lactamase enzymes documented, if you want to take that up as an area of research. These enzymes vary widely in their chemical structure and their catalytic efficiencies. And when bacterial populations have these resistant subgroups, treatment with beta-lactams can result in a resistant strain becoming more prevalent. Beta-lactam-derived antibiotics can be considered the most important antibiotic class, but they are prone to clinical resistance. And one of the reasons they're their most important antibiotic class is they're highly efficacious, they're bactericidal, they work quickly, and they have relatively low toxicity as drugs. And most of them are fairly cheap. That's another advantage. So beta-lactamases. Resistance to beta-lactam antibiotics occurs primarily, but not exclusively, but primarily through the production of beta-lactamases, which are enzymes that inactivate this antibiotic by splitting the amide bond of the beta-lactam ring. They have likely co-evolved with bacteria as a mechanism of resistance against natural antibiotics over time. That is, if you're a bacteria living in the environment next to various fungi which may be making wonderful beta-lactams, such as penicillins, having an enzyme which destroys them may be an advantage to you. So they probably have existed in the environment, these enzymes in the environment for quite some time. The selective pressure exerted by the widespread use of antimicrobial therapy in a modern medicine may have accelerated their development and spread. Now, beta-lactamases can be classified according to their amino acid structure into four large classes: A, B, C, and D. Classes A, C, and D beta-lactamases hydrolyze the beta-lactam ring through a serine residue at their active site, whereas class B enzymes are metallo-beta-lactamases that use zinc cation to break the amide bond. So the first group I'm going to talk about are the penicillinases. Natural penicillins, such as penicillin V and penicillin G, are not stable to penicillinase and have a narrow spectrum of activity. They're mainly active against gram-positive bacteria, including the penicillin-susceptible Staphylococci, Streptococcus pneumoniae, Streptococcus pyogenes, and the oral Streptococci. And when penicillin was first discovered and then used at Oxford in the 1940s, almost all staphylococci were in fact very sensitive to penicillin, and they very rapidly got resistance within a few years to penicillin with the use of this enzyme. And in our environment, only about 5% of our Staphylococcus aureus is actually sensitive to penicillin. So many anaerobic and facultative anaerobic gram-negative bacilli are resistant to penicillin. Penicillin-resistant penicillins, such as nafcillin, oxacillin, flucloxacillin, and dicloxacillin, are primarily used in the treatment of infection caused by penicillinase-producing staphylococci. Now, I don't know which particular drug you use in Indonesia. Our common ones are flucloxacillin and dicloxacillin, but you may use other drugs which have similar properties and behave in the same way. So the next group I'm going to talk about are extended spectrum beta-lactamases, and they are a serious problem and they're very prevalent. So ESBLs, which stands for extended spectrum beta-lactamases, were first detected in 1979 and are beta-lactamases, and examples are TEM and SHV, which confer resistance to extended spectrum cephalosporins, including cefotaxime, ceftriaxone, and ceftazidime, as well as the Monobactam aztreonum. The prevalence of ESBL producing bacteria have been gradually increasing in acute care hospitals. They certainly have in mine. An increasing number of plasmid-encoded ESBLs, not of the TEM or SHV lineage, have now been described. Plasmids responsible for ESBL production frequently carry genes encoding resistance to other classes of antibiotics, such as aminoglycosides. So this plasmid-mediated resistance mechanism can have multi-drug resistance. Therefore, antibiotic options in the treatment of ESBL-producing organisms are in fact extremely limited. Now, there are inhibitor-resistant beta-lactamases. Inhibitor-resistant beta-lactamases are not ESBLs, but derivatives of the TEM and SHV enzymes. They're found mainly in clinical isolates of Escherichia coli and some strains of Klebsiella pneumoniae, Klebsiella oxytoca, Proteus mirabilis, and Citrobacter freundii. They're resistant to inhibition by clavulinic acid. So these are the inhibitors: clavulinic acid, sulbactam, and thereby clinically resistant to the beta-lactam, beta-lactamase inhibitor combinations of amoxicillin clavulanate, which in this country is called Augmentin, ticacillin clavulanate, and ampicillin sulbactam. They are normally susceptible to inhibition by tazobactam, which is piperacillin, which has often associated with piperacillin, and although resistance has been described to this. So this one tends to be more stable to organisms which have this inhibitor resistance. AMP-C type beta-lactamases, that is class C. AMP-C beta-lactamases are typically encoded on the chromosome of gram-negative bacteria, including Citrobacter, Serratia, and Enterobacter species, where their expression is usually inducible. AMP-C beta-lactamases may also be carried on plasmids. AMP-C beta-lactamases, in contrast to the ESBLs, hydrolyze broad and extended spectrum cephalosporins, but are not typically inhibited by the beta-lactamase inhibitor clavulanic acid or tazobactam, whereas avibactam can maintain an inhibitory activity against this class of beta-lactamases. So this will become a more popular inhibitor agent. AMP-C type beta-lactamase organisms are often clinically grouped through the acronym SPACE, standing for Serratia, Pseudomonas, Acinetobacter, Citrobacter, and Enterobacter. So now we'll talk about the carbapenemases. Carbapenems are stable to AMP-C beta-lactamases and extended spectrum beta-lactamases. So you can use meropenem or imipenem when treating ESBLs or AMP-C type organisms. These are a diverse group of beta-lactamases that are active only against the oxyaminocephalosporins and kefamycins, but also against the carbapenems. As trionum is stable to the methylo-beta-lactamases, but many IMP and VIM producers are resistant owing to other mechanisms. And I think IMP and VIM are serine group beta-lactamases. Carbapenemases, which were formerly believed to derive only from classes A, which is KPC enzyme, B, VIM, IMP, and MDM, sorry, VIM and IMP are methylo-beta-lactamases, and D, OXA, but a class C carbapenemase has now been described. We thought we only had A, B, and D, but someone's found us one in all groups. So just to give you a little bit of molecular genetics, plasmid, that's an autonomously replicating genetic element of covalently closed circular double-stranded DNA, usually between 200 and 400 kilobases. They are extremely common in bacteria. Plasmids have a wide range of functions: antibiotic resistance, virulence, and metabolic actions. Then there are transposons. These translocate as a unit from one area of the bacterial chromosome to another, or between the chromosome and the plasmid in the bacteria. Transposons encode functional genes that mediate recognizable phenotypic characteristics. And then there are integrons, which are genetic elements that contain a site-specific recombination system able to integrate, express, and exchange DNA elements, called a gene cassette. Integrons are genetic mechanisms that allow bacteria to adapt and evolve rapidly through the stockpiling and expression of new genes. The complete integron is not considered to be a mobile element, and as such lacks functions for self-mobility, whereas the other elements I've discussed can be. So beta-lactamases, these are encoded either by the chromosomal gene of the bacteria or by transferable genes located on the plasmid or the transposons. So they can move around. This is part of the problem. In addition, beta-lactamase genes, which is often called BLA or BLA, frequently reside on integrons, which often carry multiple resistant determinants. If mobilized by transposable elements, integrons can facilitate further dissemination of multi-drug resistance among different bacterial species. So if your integron can be picked up by a plasmid or a transposon, it can take lots of resistance, not only to carbapenemase, but to other bacterial classes across into the bacteria, making them very, very multi-drug resistant. And that's part of the problem. Not just that it's a carbapenemase producing organism, but it's also carrying other resistance mechanisms. So carbapenemases, which are beta-lactamases, confer the largest antibiotic resistance spectrum because they can hydrolyze not only carbapenems, but also broad-spectrum penicillins, oxyaminocephalosporins, and lecaphomycins. The Klebsiella pneumoniae carbapenemase, called KPC enzymes, are currently the most important class A serine carbapenemases. Since initially reported from Klebsiella pneumoniae isolates, 50 different KPC enzymes have been found worldwide in multiple other gram-negative species, such as E. coli, Citrobacter, Enterobacter, Salmonella, Serratia, and Pseudomonas aeruginosa. Excuse me. Class B methylo-beta-lactamases, which are the next group of carbapenemases, use zinc cation for hydrolysis of beta-lactam and are susceptible to ion chelators such as this, which I can never say, which is called EDTA, and are resistant to clavulanic acid, tazobactam, and sulbactam. This material EDTA is used in diagnosis and identification of these enzymes in the laboratory, but cannot be used therapeutically. It's not a therapeutic drug. They confer resistance to all beta-lactam antibiotics except monobactams. So the class B metylo-beta-lactams take out everything, but as tritium. Chromosomally encoded metylo-beta-lactamases are primarily found in environment isolates of Aeromonas species, Chresiobacterium species, and Strenotrophomonas species. So these enzymes are actually intrinsic to some species of bacteria. And these bacteria will be intrinsically resistant to carbapenems. And these bacteria, fortunately, while they do cause serious disease, tend to be of lower pathogenic potential. They're not benign. So carbapenemases, the most clinically important metylo-beta-lactamases, belong to 5 different families. Imipenem, called IMP. Verona integron encoded metylo-beta-lactamase, called VIM. Zampalo metylo-beta-lactalase, SPM. German imipenemase, GIM, and Sol imipenemase, SIM. So you will see these written around the place. In Australia, we have a lot of IMP and VIM. These are typically transmitted by mobile genetic elements inserted into integrons and spread through Pseudomonas aeruginosa, Acinetobacter species, or gram-negative DON fermenters and enteric bacterial pathogens such as the Enterobacterales. So the E. coli, Klebsiella, Proteus, etc. group. The New Delhi beta-lactamase has received the most attention recently, and it's certainly increasing in Australia, its prevalence. Originally described in Klebsiella pneumoniae isolate from India in 2008, NDM1 enzymes have since been reported in a number of other countries, primarily in connection with travel to India to Pakistan. And we have a lot of people from the Indian subcontinent who live in Australia and travel backwards and forwards, and I'm sure that's part of the reason that we're seeing an increase in this enzyme in our country. These enzymes confer resistance to all beta-lactams except aztreonam. However, most metylo-beta-lactamases reside on mobile gene cassettes inserted into integrons that harbor additional antibiotic resistance genes to other microbial classes. So this multi-drug resistance can be transferred to other species via transposons and plasmids, severely limiting therapeutic options available in serious infections, as you have no doubt discovered. So the class D carbapenemases have been described among 4 subfamilies of oxotype beta-lactamases, 23, 24, 58, and I think it's 140. I can't see it. Primarily an Acinetobacter baumannii is the commonest organism that carries these. It's intrinsically weak. This is an intrinsically slightly weaker carbapenemase activity and is augmented by coupling the beta-lactamase production with additional resistance mechanisms such as decreased membrane permeability or increased active efflux, which also, independent of enzymes, can generate resistance in bacteria. So how do you diagnose carbapenemase positive bacteria? So when a bacterial strain is found to be non-susceptible to carbapenems or have either intermediate or totally resistant, the mechanism of resistance is still actually unknown. It could be decreased affinity for the antibiotic binding site, i.e. the penicillin binding protein. It could be enzymatic, e.g. the carbapenemase destruction, or it could be decreased concentration of the antibiotic inside the bacteria, such as porins, so the bacteria can't get in, or efflux, so the bacteria keeps pumping the antibiotic out. So there are various mechanisms of resistance to carbapenems, which can be a problem. Thus, to confirm the production of a carbapenemase or the presence of other mechanisms, further biochemical assays and or gene-based tests need to be performed on the isolates. And determining the mechanism of carbapenem resistance can help in the selection of the most appropriate antibiotic therapy early in the treatment of gram-negative infections. Testing for carbapenemases can be phenotypic and or molecular detection methods. So fundamental considerations. A negative test does not imply that the organism is carbapenem susceptible, as it may still be resistant due to non-enzymatic mechanisms, such as efflux and porin issues may have a thing, or just binding to the penicillin binding proteins. Conversely, the presence of a gene does not systematically imply the organism is carbapenem resistant, owing to the level of expression of the resistance gene. So if your carbapenem resistance gene doesn't have a very good promoter and the enzyme and the gene is not expressed and there's not much enzyme made, again, you won't see resistance even though the organism has this gene. So a positive biochemical test will not identify the specific carbapenemase enzyme, but a molecular test may overcall, because you can detect the gene, may overcall true carbapenem resistance. So at the end of the day, what counts is phenotype, okay? So consequently, only phenotypic tests rely on actual growth inhibitor to provide a full susceptibility profile picture for you. So while molecular testing is very helpful and very quick and gives you a good idea of what's happening, it doesn't actually necessarily give you the whole answer. Currently, the antibiotic options for the treatment of carbapenem-resistant Enterobacteriae are very limited, with polymyxin, tigecycline, fosfomycin, nitrofurantoin, and some aminoglycoside as the mainstays of therapy, which you may still be able to use. And that will depend upon what other classes of antibiotics are actually carried on the plasmids or transposons, which are carrying your resistance genes. So here's just an outline, a brief outline, looking at different antimicrobials. So Astrianum avibactam, cefiderocol. Both of these are very, very expensive. Ceftazidime avibactam, very expensive. Ceftolozane tazobactam, very expensive. Evercycline, fosfomycin, imipenem relbactam, meropenem vaboribactam, plasmamycin, polymyxin B or colistin, which are probably quite cheap, tigecycline. And this is just giving a rough indication of what we would expect to work. So red, don't use it. Green, you have a good chance that it may well work. And yellow, it may or may not work. So this table is quite useful very early on to decide. The trouble is that all these drugs, most of the drugs down here, are in fact extraordinarily expensive. In Australia, they would be thousands and thousands of dollars for a course of therapy. I mean, thousands and thousands and thousands of dollars. Very, very expensive. Okay, so a good thing to stay away from. So in conclusion, carbapenem-resistant gram-negative pathogens have become a major healthcare burden in the 21st century, not just in Indonesia, but globally, and they're certainly causing increasing problems in Australia. Treatment options have been limited to agents such as colistin, which is a very old antibiotic with significant toxicity. Tigecycline, which is a newer antibiotic, has limited therapeutic efficacy depending upon penetration and where the drug can get. And we tend not to use this class of drugs in young children, although if it's about saving someone's life, one would use it. Fosfomycin, again, is a fairly limited activity and is mainly active against E. coli and not much else. Nitrofurantoin, again, not a very strong or effective antibiotic, particularly and mainly used in urinary tract infections. But these can be then used in combination with other antibiotics if there are other antibiotics you can use. Fortunately, several new agents with activity against carbapenemase resistance have been approved or in the late stages of clinical development, which is encouraging, but as I said, extremely expensive. They may be a bit cheaper for you in Indonesia than they are for us. That's about drug companies just charging what they can get away with. These newer agents will become important additions to the current limited options and are expected to improve the outcome for patients infected by carbapenemase resistant pathogens. But watch this space. I'm sure they'll develop resistance to those too. And most importantly, antimicrobial stewardship and infection prevention and control play a crucial role in ensuring the optimal and rational use. So the infection prevention and control is very important to keep these patients who are colonized or infected with these organisms separate from those patients who are not, so that you don't have a much higher prevalence of problems. And antimicrobial stewardship to guide in the appropriate use of antibiotics, and particularly in stopping the antibiotics when you've achieved a result, is very important to minimize exposure of these organisms to these drugs so that they will have a longer life and availability to us before we get resistance, because they probably, time will tell, I'm sure we will get resistance. We have to most other things. Okay, so that's my talk. Thank you very much.
Thank you very much, Professor Alison, for your presentation. It is very precise, but also very deep essay. Information is also comprehensively combined with treatment and also some prevention, but it is still in an outline. So, for the next step, we will start our discussion session. We will have 22 sessions for the discussion. For the first round, I would like to invite maybe 2 or 3 participants to ask questions. You can ask directly, or you can also write in the chat room in Indonesian language, or also in English language. So, but we can accept this, and we will read this for Professor Alison. While waiting for the participant question, I would like to ask you, Professor Alison, regarding with the prevention. Yeah. So, if there is already, so our hospital, many hospitals in Indonesia now already experience already face a lot of infection caused by Carbapenemase producing Enterobacterales. And if, for instance, can we reverse this resistance by, for instance, stop using carbapenem and change it with other non-carbapenem? For instance, what is said as carbapenem antibiotic cycling strategy like this? Can we do that, and then can we reverse the situation with high prevalence of carbapenem resistance to fewer to lower resistance to carbapenem?
There is some evidence using in other antibiotic situations that doing that and decreasing the use of certain classes of antibiotics decreases the prevalence of resistance, such as controlling the use of third-generation cephalosporins has had some impact on decreasing the level of resistance to these drugs. But it's difficult to do. They're also one of the problems, though, of the carbapenemases, as I was saying earlier, is they often carry other multiple resistance genes to different classes of antibiotics on their plasmids. So you you lose all your beta lactams, you lose aminoglycosides, you may lose macrolides, etc., or quinolones particularly, because you can rapidly get quinolone resistance just independent of this. They're intrinsically easy to get resistance to. So that becomes a problem, is what are you going to, if you're not going to use carbapenemase, what are you going to use? And that will depend upon your local epidemiology, what you've got. The things that you also have to consider are, where is the likely site of infection, and will the drug penetrate to that site of infection? Such as macrolides, you can't use very well in the brain. Aminoglycosides only get into the brain fairly slowly and are not great if you've got gram-negative infection in the brain, which fortunately is very rare, except in neonates. So you have to consider where the most likely site of infection is, the penetration of the agent, and its efficacy in working, such as, for instance, this is not a carbapenemase problem, but if you've got Staph aureus endocarditis, which is an MRSA, but sensitive to erythromycin or clindamycin, you'll probably get failure if you use those drugs. They just don't work very well for that particular infection, and you need to use vancomycin or something, something else, but certainly not macrolides. So a lot of that will have to do with that sort of level of issues. There is also, for instance, another issue in the urine, but all the antibiotic sensitivity data that is given out correlates with achievable blood levels that you can get in serum and in blood of antibiotics, and thus we're making an assumption that that will be a similar level of concentration of drug in certain tissues. Now, getting across the blood-brain barrier can be a problem, and so some of those drugs, such as aminoglycosides, I don't think you get very good levels in the central nervous system. Alternatively, in the urine, you can sometimes get very high levels of drugs, much higher than you have in the bloodstream, which will mean that those breakpoints of deciding sensitivity and resistance, you may still get above it and be able to eradicate the organisms, such as Enterococcus faecalis, which is ampicillin resistant. You could still often treat in the urine with oral amoxicillin or intravenous ampicillin, because you get so much in the urine, it can still be efficacious. So there are some other things to consider when choosing therapy and what you're doing. But certainly, I mean, if we took away all the antibiotics and didn't use them, I'm sure a lot of the resistance levels would eventually become much lower. Of course, we'd also have a lot more people die from infections, which would be the problem. So it's a difficult issue to balance between withdrawing or decreasing the use of antibiotics and selecting something that's rational and sensible, and using as much as you need, but no more. I hope that's helpful. It's not an easy answer.
Yes, so it is maybe probably it is can be done for urinary tract infection, but for other infection, maybe it is not easy, because not it is difficult to find a bacteria antibiotic which can replace meropenem for certain infection.
Yeah.
Okay, so now we have already some questions from the participants. This is from Dr. Afandi Bagus from Medical Microbiology Specialty Program from Malam.
What should we use?
Dr. Fahri from Dr. Would you please show me the question list? I need to read the question list, yeah, okay. So the question is, does carrying carbapenemase resistant gene make people traveling between countries require special screening? How can we prevent and control it?
Well, currently you don't need to be screened for much to get into Australia, not as a visitor. To emigrate into Australia, to come here to live permanently, you get screened for tuberculosis, but then you just need to be treated, so you don't come in with active tuberculosis. But we know we don't, in this country, we certainly don't do this. The trouble is how you would do it, and you would also have to legislate for it, because I don't think you can do this without it being entrenched in your legal system. At least not in this country. You couldn't do this without it being entrenched in your legal system. But I'm sure we're all carrying resistant bacteria of some sort or another, so I'm not quite sure that this is going to solve your problem. I think one of the really important things to do, though, is to emphasize to people to wash their hands. You know, hand washing, so you don't, and in hospitals and just generally in the environment, I think is very important. So I don't think it'd work. I mean, yes, if no one left Indonesia and no one left Australia, we wouldn't take our resistant organisms elsewhere. But I don't think that's going to be very acceptable to the societies. So that's a problem. But can I recommend hand washing and that you encourage your colleagues to do it? And by the way, the doctors are always the worst at hand washing in terms of doing it. Wherever you go, certainly in Australia and anywhere else I've asked, it's always the doctors the worst. So doctors, wash your hands.
Yeah. So screening for carbapenemase resistant genes is probably not very applicable this moment, yeah. But it is more important to implement the transmission prevention by by like standard precaution like hand washing and so on. And okay, so now the set condition from Dr. Fahri from microbiology specialty program at Ponorogo University. What should we use to clean waste or surrounding environment so that it's free from the genetic materials of Carbapenemase resistant bacteria? And how do we ensure that our environment is properly cleaned so that it is free from CRE carrying genes that can be transmitted through horizontal gene transfer such as plasmids?
Okay, so it depends what we're talking about as the environment. If you're talking about the general world and going out, walking in the parks with the trees and the plants and whatever, I don't think it's possible, right? Because they're just part of the normal environment. So now let's assume the environment we're really interested in is the hospital and the hospital issues, and to keep the hospital environment as clean as possible for all these very reasons, so that we don't have lots of bacteria with resistance genes, be they carbapenemase or others, so that can be transferred on people's hands, which is the most common way, from person to person. That's the hand washing. So first of all is that you can go around your hospital and you can scrub it with disinfectants, various types of antiseptics or disinfectants. But the disadvantage of that is that you will then find that your bacteria you'll select out the bacteria that are resistant to these chemicals, because that happens quite a lot. And so you won't ever generate a sterile environment. So cleaning is clean it so it is physically clean. There is no dirt or grime or blood or secretions or things. So the first thing is to get it physically clean. And with that, I would use neutral soap and detergent. And I would not be using any disinfectant type materials unless there was a very special reason to do it, because I think you will find that you will just end up with your environment colonized with organisms which are resistant to your disinfectant. It's no different to antibacterials. So I think the most important issue to bring up in this is, yes, you need a clean environment, and that means cleaning, but using neutral detergents, not chemicals to destroy them. There are some chemicals which will kill everything, but they're very toxic. Things like bleach will. Obviously, very high temperatures will. Sorry, I'm getting emails to do things. There are things that you can do, but they are fairly toxic and not very practical. So I think cleaning, yes, and hand washing, yes, so you don't transmit it, and or wearing in a highly infectious in an area where there are infected people, wearing a gown that you can take off and leave behind and not walk it around the hospital, is good. But using chemicals, I would use very sparingly.
Is there a specific antiseptic or disinfectant that
For
to be
carbapenemase, no,
implemented?
you're not going for the carbapenemase, you're going for the bacteria. So it's whatever kills out the types of bacteria. And Enterobacterales are not all that hard to kill, they're in a physical sense. Bleach is very good, so chlorides are very good. But chlorhexidine is good, but it doesn't cover Pseudomonas. is a good on for hands, you know, so you have alcohol hand rub with chlorhexidine. That keeps down a lot of bugs, but not Pseudomonas. The trouble is when you take away one group of bacteria, another group will grow up. You will not get a sterile environment. So you need to remember that. It may sound like a great idea, but nature does not let you have a vacuum, and nature will fill up that clean space with other microorganisms.
She agree about an outline about the importance of hand washing is the important way to prevent infection. So now we can continue to the next question. There are one audience that raise hand to ask question. Dr. Elizabeth Edwina from Diponegoro University Semarang, please.
Thank you, Dr. Helmia. Thank you, Prof. Allison, for your great lecture from Undip. Prof. Allison, is it possible that non-ESBL Enterobacterales are resistant to carbapenems, or we said it CRE, carbapenem resistant Enterobacterales, as in the case for Pseudomonas aeruginosa?
Well, Pseudomonas
is it possible that we find an Enterobacterales that resistant to cephalosporin third generation? Thank you.
Sorry, resistant to what?
Allison.
Third generation cephalosporins? What is?
Yes, Prof. Allison, is it possible that Enterobacterales that resistant for Carbapenemase but still sensitive for cephalosporin third generation?
Yes, but it won't be by carbapenemase. It will be because of efflux mechanisms or porin issues in your organism. So either the antibiotic can't get in the first place, or the organism is rapidly pumping it out through efflux. But it won't be because of the enzyme, because carbapenemases, essentially, if you like, super strong ESBLs in a way. They do everything an ESBL does plus more. It's not as if they just take out imipenem and meropenem. They will take out all the beta-lactams, every one of them except for probably astrenum, depending on the class. So it's, yes, you can get that in Pseudomonas, but it's because of other mechanisms of resistance, not from the carbapenemase. But that is important too.
Okay, thank you, Prof. Allison.
Okay, thank you. So it is possible, Prof, there is can be found in anti-diagram. An isolate is probably resistant to carbapenem, but it is still sensitive to cephalosporin third generation. Yeah, it is possible still to find this, but yeah, because
the answer, I'm not sure. I wouldn't like to bet on it. In theory, I can say yes, but I'm not certain that the answer to that is. Helmia, I'm sorry.
Okay, thank you. And is that another question from participants? I would like to invite that. Okay, there is a question from Dr. Anna. She is from Diponegoro University. So she asked about, do we have to do a confirmatory test for carbapenemase every time we get an AST result that is resistant to one of the carbapenems on the automatic machine?
Well, the answer to that will depend upon what system you're using for antimicrobial sensitivity testing. And in my laboratory, we use the UCAS system. And so it's actually screening on a specific sensitivity, specific disc on specific agar, like Mueller-Hinton blood or whatever we do our sensitivity testing on. And I can't remember the strength of the disc for meropenem, but it's in our manuals and it's what we use. And if you get a certain, if the diameter is too small, those in our laboratory are automatically, then we go on and test. But I don't know whether you have to do it. I'm not in Indonesia. I can't tell you what to do. And I think you probably use the CLSI method which is fine. UCAST and CLSI are both excellent international methods. I'm happy with either of them. I'm also happy with the Australian one, CDS, but that's fine. So whatever method that you're using, you need to follow the rules of the game of whatever they're telling you to do, you need to do it, and you need to do it exactly as they say if you want a reasonably accurate result. None of these results will be perfect. You will still very occasionally call the sensitive organism resistant and a resistant organism sensitive. No system is perfect, but you need to have a good international system which has a lot of data and information behind it to support what they're saying, and follow the rules very strictly of whatever they're telling you to do. And the basic answer is, if I if my screening thing was suggesting that the patient was a carbapenemase producing organism, yes, I would be doing the screening tests, and I would be withholding that result from clinicians until I knew the answer. And I would be strongly suggesting they didn't use a carbapenem until we knew that it was safe to do so for that patient, and try and suggest something else for them.
Okay, thank you. As there is another question from a participant asking from University Indonesia. She asked about in hospital setting, is screening important? Is screening important to find colonization of CRE in patients, and how we select the patients to screen? Is there any recommendation?
Okay, yeah. Now, there is only any point, the question you have to ask is, why are you screening? And if you find a positive or a negative, what are you going to do about it? And if you're not going to do anything about it, then there's not a whole lot of point in doing the screening in the first place, right? That's your first thing. So you've got to think, what are you going to do? So the reason we do not routinely screen patients, well, actually we do, because we've got a high risk situation here, but it's up to you. So say you do decide to routinely screen. So what we do is, if we get a child who's an inpatient and we get a fecal specimen sent in for, have they got diarrhea from rotavirus or Campylobacter or something like that, we will also go and look to see whether they're carrying VRE, ESBLs or CPEs for infection control purposes. And most of them don't, but we do pick them up. And then that patient gets put under infection prevention and control, very strict contact isolation precautions. If we found a CPE, any of those organisms, they would be under very strict infection prevention and control precautions. So we do that in a sense here, because it's a tertiary pediatric hospital with a lot of transplant patients and other very, very sick children. So you need to think about what you're going to do and what you can do about it. If you can't isolate them or do anything, you need to be able to at least flag them and tell people to wash their hands and wear gowns, and take their gowns off and then wash their hands afterwards. So then the other issue when you screen is, if you've got a relatively low prevalence and you pick up a patient who's infected with a CPE who's in your hospital, should you screen the patients who share the room with that patient or nearby? And that you need to decide. So it's best, if you're going to do any screening for ongoing transmission, is starting close to the patient and screen the ones that are immediately around the patient. And if you don't find any there, you could decide whether you want to spread out further. If you do find in the first room that there are other patients with that organism, you may want to look in other rooms or areas of your hospital, particularly where some of those patients may have been. So it becomes very difficult and hard to do. It's a big burden on the laboratory, and they need to be aware of it so that they've got plenty of agar and available to do the screening and test and look. So you need to think carefully about what you're going to do, who you're going to do it to, and what your goals are. And I think you need to have a discussion with your infection prevention and control and laboratory staff, and decide on a strategy of approaching this problem. If a patient is colonized with CRA, how would you treat them? Well, you don't need to treat colonization because they're not infected. Don't treat them at all. You only need to treat infections. You cannot clear colonization. Okay? If you take away the antimicrobial pressure, so if the patient's sick in hospital and on antimicrobials, they've got pneumonia with pneumococcus, and you're treating them with penicillin and they're getting better, or you're treating them with cefotaxime because it's a bit of a resistant pneumococcus or something and they're getting better, you may well find, if you screen their feces, that you would find CPE, but once you take away the antibiotic pressure, that the organism will still be there, but it'll be in much lower numbers and harder to detect in the laboratory because of the number issue, etc. So, one, I would not treat anyone who's colonised. I would note on their medical chart that they are colonised. So, if they came in with what looked like very, very serious gram-negative sepsis, that I would know that starting with a carbapenem may not be the greatest choice to save their life. And it may also be important as to where you place them in your hospital if they return in terms of infection prevention and control. But that's what I would do with colonisation. Do not treat it. You cannot cure it. Not that we know of yet, anyway. So is that okay, Helmia? We seem to have run out of questions, which is fine.
Oh, sorry, yeah, sorry. So there's no specific strategy for instance, for patient which is colonized by CRA, ya, Prof, ya. Not unlike the patient which is colonized by MRSA. In some hospital, they use like mupirocin to control the colonization, but there is no specific strategy for CRA, Prof. Is that correct?
Sorry, run that past me again. Say that again.
Yeah. Yeah, so there is no specific measures to control colonization or carrier of CRA, Prof. Yeah. It's unlike in maybe some hospital, they have a policy to control MRSA colonization using mupirocin, but this is not it doesn't work for CRA, something like that.
There are methods to allegedly decolonize people who have MRSA, and that has been done. And
Yeah.
the trouble with that is I think that they just get themselves recolonized when they go out in the right world and meet the bugs in again anyway, just because they're people who tend to be colonized with a third of the people are walking around with Staph aureus in their nose. And a certain percentage of those will have MRSA by virtue of the fact of whatever's in your environment. There is no easy way. Well, there is a way that you can screen for carbapenemase resistant Enterobacteriaceae. There are chrome agars which will do it, which makes life a bit easier because of looking for color changes. You could think of the otherwise it becomes a very difficult to sort of going through all sorts of gram-negative organisms and trying to figure out which one may or may not have the resistance gene. So it would be an awful lot of work for not a lot of yield and very expensive. You could try, but I don't know if this works, is just, having mixed fecal culture and having a meropenem disc and seeing what grows within a certain zone and testing those. But you will find a lot of bacteria because there will be a lot of things that are intrinsically resistant. I've already told you, Crasiomonas, Aeromonas, Stenotrophomonas are all intrinsically resistant to carbapenems because God made them that way. That's how they are. So that will occur, and that's not such an infection control problem because that's chromosomal, and that's just the nature of the bacteria. It's the ones that have plasmids and transposons where this genetic element can jump from one bacteria to another. The other problem with carbapenemase resistance and tracking it in hospitals is you're not necessarily tracking a bacterial species such as vancomycin-resistant enterococci or methicillin-resistant Staph aureus. You're chasing a plasmid, and that can be in anything. It can be in E. coli and Klebsiella and Citrobacter and Proteus, Pseudomonas, etc. So it becomes a lot more difficult. You're not just going after a species. It could be in any number of species. So we screen using chrome agars, and if we pick up something or if we happen to see something on a disc and sensitivity testing that we think fits within the zone for meropenem, then we go on and do phenotypic screening. And we can do a quick genetic test, which is on Gene Expert, which does a lot, but not all of some of the genes, so that that helps you to know whether what you're seeing is real. Like I said, there could be issues that you find the gene, but it's not expressed, so it's not a risk. So it's a combination and a decision. You would have to sit down and have a hard think about what you're doing, what you're trying to achieve, and how you're going to do it, and what resources you have to do it. We're very resource constrained, like everywhere else, and I'm sure you are too. Is the number of people, the amount of money all this costs. But just growing, streaking feces on a plate and trying to find the resistant gram-negative, I think will be very, very, very difficult amongst the zoo that you will grow.
Okay, thank you. And the next question, can you please show the question list? There is still 2 more questions, I think.
Okay, then I have to go to work.
Yeah, and no, yeah, but we still have maybe 10 minutes to this discussion, if you don't mind.
Okay, which patients are at increased risk for carbapenemase resistant Enterobacteriale acquisition? Those patients who have multiple exposures to a high prevalence environment, such as a hospital, right? So if you keep going in and out of a hospital where you're going to find more of these organisms, you're at risk. If you are somebody who gets a lot of antibiotic exposure, such as cancer patients who get fever and neutropenia get a lot of antibiotic exposure, they are more at risk because you end up selecting for the resistant organisms resistant to the antibiotics that you put on. So those are the patients that I think are significantly at increased risk, or those who get long exposure to antibiotics, who have antibiotics for a long period of time for whatever issue. So if you don't need it, stop it. Stopping getting doctors to stop therapy is always the hard part. So those things I think put them at risk. One is the environment, going into a high prevalence environment, and the second is antibiotic exposure. When can contact precautions be discontinued for patients colonised or infected with these organisms? The answer is at the moment, never. I do not know of any reliable mechanism that has been proven that will eradicate carriage of this group of organisms. So there it goes. There's no reason why you can't cohort them though. So if you've got 3 people with this sort of resistance, there's no reason why you can't put those 3 in a 3-bedded room away from other people. They don't all need single rooms. It would be ideally wonderful if they all had exactly the same plasmid, but you're never going to be able to sort that out. That would become very, very expensive and difficult and time consuming. But you can at least think about cohorting them, but they need to be within that room. You still need to have contact precautions, and that people still need to be wearing gowns, and there needs to be at least 1 metre between the beds, and there needs to be very strict hand hygiene, and those patients are not to share things amongst themselves. Okay, but you could certainly cohort them, put them all in the same room. In a sense, I mean, ideally it'd be wonderful they all had their own individual room with their own individual bathroom, etc. I don't know what your facilities are like. We certainly don't stretch to that. We don't have that facility available. So we sometimes have to put at-risk situations together. More likely for us it's ESBLs, but certainly carbapenemase type problems would be exactly the same issue, or VRE, and for that matter, MRSA.
Yeah. Okay, thank you. So there is a participant who raised hand. Dr. Rebrenda, please.
Yeah, thank you very much, Dr. Helmi, and also Professor Kesson. I had sometimes isolates that is only sensitive to amikacin or chloramphenicol in the AST result. So if we assume that both amikacin or chloramphenicol can enter the site of infection in a good concentration, do you have any experience or views about giving chloramphenicol or amikacin only for patients with CRE infection, Prof?
We don't have chloramphenicol available in Australia except under a special access scheme because of the toxicity issues. However, when I was a junior doctor, we used it a lot, and it's a very good drug. It is fairly broad spectrum, and it is a static agent, not a cidal agent, but equally it has been used and it does cure. And we used it for beta-lactamase producing Haemophilus influenzae meningitis, which was, when I was a very junior doctor in the very late 70s, early 80s, was a very prevalent problem until vaccination came in the mid-90s. So I think chloramphenicol is a very good drug, but it does have a problem of bone marrow suppression, and that limits the duration of use. I don't think you can really push chloramphenicol beyond 1 to 2 weeks of therapy without seeing a significant drop. And once you start to see a drop in white cell count, and you need to be following it closely, if you have 2 days in a row with the white cell count going down, now it can go down because the infection's improving, right? As the infection gets better, the white cell counts. But if it's falling and you're concerned about it, you need to stop the drug because it will continue to fall. And you do not want to make the patient aplastic because that'll make your problems even worse. So chloramphenicol, I personally think, is an excellent drug, has really good penetration into tissues. It has very broad, good spectrum. It doesn't get used much, at least in my country, so there will be probably less resistance to it. So there's that. But the toxicity, the big toxicity is bone marrow suppression. And it's for real. I've done that. I've dropped people's white counts and sat around very nervously for a week or two waiting for it to come up. And I've used it to treat brain abscesses in America, and it worked very well, but one has to be very careful with it. Then there's amikacin. Again, I don't know what it's like in your country. That's actually a fairly expensive drug. It's a lot more expensive than gentamicin in my country. But it's a very good drug. But again, it has penetration issues, and it depends where and what you're trying to treat. If it's in the renal system, yeah, it's a good drug. If it's in the brain, no, it's not a very good drug. If it's in the bloodstream, it's probably a very good drug. Pneumonia, I've never used amikacin for pneumonia, but I would think that would work well because the lungs have a very good blood supply, clearly, because that's how the oxygen gets into your body. So they've got a good blood supply. So I think that's true. But again, it's a toxicity issue of being deaf, vertigo, so vestibular problems, eighth nerve deafness, and also renal failure would be the big problems. And so monitoring the therapy. Now we do have drug level monitoring. I don't know what's available, not in every hospital in Australia for sure, but certainly in this hospital where I work, we could monitor it fairly effectively. I don't know what's available easily in Indonesia. So again, it's a toxicity issue. So it's always a measure of efficacy of the drug, toxicity of the drug, cost of the drug are the things that we're always considering. You know, if I can use a cheaper drug that's just as effective, I'm not going to use the expensive one. The other thing is that a lot of doctors think broad spectrum antibiotics are better. And broad spectrum just means it kills a wider range of type of bacteria, as you know. It doesn't mean it kills them any better. It's not better. One of the best drugs we have is, in fact, penicillin. If you want to kill group A strep, penicillin is the drug to go for, not meropenem. You know, so people also have this misunderstanding that the newer drugs and the broader spectrum, oh, they must be better. They're not necessarily better at doing the job that you want. It's their spectrum of activity or their resistance to other situations. So is one better than the other? The answer is I don't know. They both carry their risks. They're both efficacious. They both have toxicity problems. They both have penetration and getting to be, while they're intrinsically efficacious on the disc, to be efficacious in the patient in the circumstance that you're treating. And then there's the cost issue, which is also important, because if you spend the money on that, you can't spend it on something else. That's how it goes. Yeah. I'm sure your hospital budgets are finite, just like ours are. There's only so much money to go around, and once you've spent it, you've spent it. Just like your home salary. Once you've spent all your money, you've spent all your money. That's it.
Yeah.
You got the same problem.
Thank you. Maybe another question, Dr. Helmia, if it's okay.
Yes.
Yeah.
Yes, please. Okay.
Professor Kesson. So I met this kind of situation many times in my clinical practice. So if a patient intubated with infection, but we treat with antibiotics and the patient improves, so no more sign of infection, the procalcitonin is going normal again, but the patient is still intubated, cannot be extubated. But the clinicians very often hesitate to stop the antibiotics. Do you face also this kind of situation in your clinical practice, and what do you suggest to this doctor? Thank you.
Well, the issue is, getting people to stop when the patient's getting better is always a big deal. Like I said, I think we probably all go on with a bit longer than we need to with antibiotics, including me, sometimes, and it's out of nervousness and wanting the patient to be safe. There are things that I consider. First of all, in treating an infection, you actually only have 3 mechanisms you can do. You can remove the foreign body, like if it's an endotracheal tube, or if it's a piece of wood stuck in your leg from a fall over and an injury, is remove the foreign body. Because until you do, you cannot cure the infection. So it'd be nice to take the tube out, but obviously you can't. That's one. Number 2 is drain pus. If there's lots of pus, get it drained, because that's just dead tissue that you cannot get antibiotics in to kill bugs. So surgical treatment of infections as part of it is very important. And sometimes in some minor abscesses and things, that's all you need to do is just drain them. You don't need antibiotics. So remove the foreign body, drain the pus, and finally give the right antibiotic, not the wrong one. So that's all you've got to do to treat an infection. Now, in terms of taking the tube out, then you can have a discussion with them about the risk is if they continue the antibiotics, that's terrific. And the patient probably won't get infected with that organism again, but it's like anything else. If you give antibiotics and you eradicate big families of bacteria, other things will grow in its space. So if it isn't bacteria, it'll be yeast, right? So you can't make it sterile, short of sticking the patient through the autoclave, which won't work. So you can't make them sterile. So it becomes a risk-benefit thing. So the longer that you keep doing this and giving antibiotics, the further you're going to chase down the resistance tree, because for sure you're going to get whatever you get colonized with is going to be resistant to the antibiotic you use, right? So then you're going to go to the next. So you're going to go to the next one, to the next one, to the next one. So you're going to get to the end of the line. The next thing is, what's the risk? So if the patient does get pneumonia again, are they going to sort of drop dead in 24 hours, or will you have a day or two to get on top of it again if this happens? Because that often is something I think about when I'm approaching a patient. If a patient's got septic shock and looks like they're about to die in the next 24 hours, you'll go in with all guns blazing and with, really heavy duty antibiotics. If they come on with an infected toe, if you get the antibiotics wrong for a day, your empiric choice is wrong for a day or two, it's probably not going to be a huge ongoing damage to the patient. So there are those sorts of considerations to take into place. But I think people need to understand a lot of the time they're treating the patient, they're treating themselves for their own anxiety. They cannot make the patient sterile. So if they give the antibiotic, that's fine, but the next infection will be resistant to whatever they're using, and there will be a next infection if they keep doing this. They won't stop it. And I guess finally, it's the risk of recurrence. If the patient does get a bit more ventilator-associated pneumonia next week, can you manage it again? It'll probably, if you've stopped the antibiotics, it'll probably be the same bug again. If you keep going with the antibiotics, it'll be a different bug. So I would put that to them. They need the confidence to try it. They need the confidence to try it and see what happens carefully and in a safe environment. If the patient's critically ill, well, they'll have to judge that.
Yeah, so it's we need to really work hard about this, because many doctors still not confident enough to appropriate antibiotic according to the result culture, also according to even the clinical improvement. We still want to continue. Yeah, so we need to educate this, and if this doesn't work, so we need maybe the power of system, like what the department and the Ministry of Health already make a policy about PGA, penatagunaan antibiotik gitu ya, antibiotic stewardship program. Because as only what I observe in Australia, in Sydney, Prof, in your hospital, almost all antibiotics are given based on infectious disease expertise, Prof, and the doctors really comply with it.
Well, I think only half are, but anyway, we do try, and we do not only those that we are formally asked about, but in other situations that become aware of in the laboratory, we will talk to the doctors about, or that we become aware of, we will talk to the doctors and try and talk them down off things, but it can be difficult sometimes.
So yeah, okay. Thank you, Prof. But we the time is almost over, but I still have one question. Maybe this is the last question. So what kind of antimicrobial stewardship program that should be properly would you suggest to us to prevent the increase of Carbapenemase producing Enterobacterales in our hospitals?
You need to, it won't work unless you manage to engage your clinicians to trust you. It won't work as a dictatorial system, at least not in this country. You can't go in and dictate. They're not my patients. I'm not responsible for them. So they have to get their trust. So explaining to them what you're trying to do and why, trying to give them alternative approaches, making sure that you're available to answer questions, we have, a structured antimicrobial use. So there's those antibiotics which we don't restrict. In reality, don't tell anyone, but I don't restrict any antibiotics. What I do is make them justify them if they start them, because I'm not going to be seeing all these patients. There's hundreds of them. I can't see hundreds of patients in a day and figure out what's wrong with them and what antibiotics they should have. But if we become aware, we have a watch list. So if you've had a watch list of all patients on carbapenems and then went round and assessed whether you thought this was justifiable or not justifiable, and also once you've got laboratory results that people actually promptly change away from carbapenems if they can, then I think those are the sorts of things. But I think it's got to be collaborative and they've got to trust you. And it won't work without those two things. So after that, how you actually go about it is up to you. That, and also getting people to stop when they don't need to. There's very few situations that you need to go past 2 weeks of antibiotics for treating an infection. There are some endocarditis, some cases of endocarditis, clearly pneumocystis, prophylaxis, osteomyelitis, brain abscesses. There are some situations where we go past 2 weeks, but patients with pneumonia and urinary tract infections and things, wound infections, 2 weeks should be plenty. More than enough. In fact, in most cases, more than enough. Too much.
Okay. Okay. Thank you very much, Professor Allison.
You're welcome.
Okay. So no, the time is already over, yeah, for the discussion.
Yeah, I need to get back to my work too.
So, okay, thank you very much. I would like to make a closing statement, maybe as a conclusion that Carbapenemase producing Enterobacterales is a serious problem in Indonesia. So we really need to be careful about this, to do something that is intensively to overcome this. An antimicrobial stewardship program, which is including many parties, should be started and also performed consistently, and also infection prevention, which include hand washing, should be consequently implemented. I think
Absolutely.
This is the conclusion statement. Yes. Okay. Again, thank you very much from us, Professor Allison, from your time and your lecture. Hopefully this will be benefit for all us. And also thank you very much for the participants for actively participate, give questions and also comments on this discussion. Again, so thank you very much. Yeah, thank you. And I give the screen back to the MC.
Okay.
Assalamualaikum warahmatullahi wabarakatuh. See you, Professor. Bye-bye.
Farewell.
Thank you.
Bye.
Thank you. Thank you, Professor Kesan and Dr. Helmia. The presentation and discussion were extraordinary so that we can get a lot of knowledge to apply in everyday life. Many thanks to speaker, moderator, and participant who asked and listened to the event from start and to finish. On behalf of the committee, I apologize if there is something that did not go correctly. Finally, thank you so much. See you in the next guest lecture event, and wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih teman-teman Presiden, terima kasih Dr. Helmia, terima kasih Bapak Ibu semua.
Terima kasih dokter.
Terima kasih dok.